Okay, so we're going to kick off today by talking about microstructural and histological features. But before we even start doing that, I wanted to talk a little bit about why. So why would we parcellate the brain? And this year, educational courses are supposed to be interactive. So you guys can either shout at me and I'll repeat what I heard, which could end up being really confusing depending what I hear, or the microphones are on. So why? Why would we bother to parcellate the brain? Anybody? Why are you here? Okay, I guess I'll talk to myself. So, one possibility to localize function. So no points for any of you because that was actually in the title of the session and all you had to do was open your session books. Another possibility, and I think this is a really important one, is to create a common language to understand and talk about the brain between ourselves. So if we have something going on and we don't know how to describe to somebody else what's going on, then how are we ever going to figure out what's happening in the brain? And maybe another possibility is that we want to understand how individual brains vary and differ from each other and are similar to each other. And I'm not just talking about one human brain to another human brain here, but also the brain of one species to another species. So I think these are some of the key concepts, and I'm sure you guys can think of more, even if it's a little too early to yell them at me. And so there are multiple levels of anatomical parcellation that we can think about. And the one that's most obvious to think about and easiest to do is to look at the macrostructural gyral and sulcal variability in the brain, which is shown in the top left right here. And one thing that has become really clear about this is that it's pretty variable. So here are parcellations of a couple of gyri here across multiple subjects. Trying to get the mouse back there. And what you can see is that there's a lot of variability across those measures. And there's a possibility here that this isn't the best measure for us to try and line things up from one brain to the next. And so because this is a session, I'm not going to talk about all of these levels, but I'm particularly going to talk about cytoarchitecture, myeloarchitecture. These are names for the microstructure of the brain. And other speakers in the session will focus on things like connectivity and function as measured by MRI. So the next question then is why use microstructure? So I'm still going to ask you guys to talk to me even if you refuse to do so. So why might we be interested in microstructure? Other speakers?
The category of cells can be related to the kind of function that is processed.
Right. Right, so different cell types might have different functions or be related to different functions.
Also the circuitry, the local circuitry of a cortical area is going to be best reflected in its microstructure.
Right, yeah.
Processing is it doing?
Yes, I don't know if you heard that, but the local microcircuitry is going to be so the local connections between and within brain areas is most accurately going to be represented by microstructure. And again, that might have a knock-on effect on function. Also, that I just said that macrostructural landmarks can be problematic, and they certainly can't capture those aspects. So not only are they variable, but they can't actually capture those more local aspects. In addition, some people have argued that microstructural maps are really objective. So they really are repeatable and precise, and we can come up with really great criteria for them. This isn't to say they're better than anything else, but that they have that feature. And in addition, as we said, it might relate to the functional organization of these areas. And some of this work goes back a really long way. So these maps are some of the earliest to consider the link between structure and function. This is from Vogt and Vogt in the early 1900s, in the 1920s. And they went through human brain slices post-mortem and looked at the cellular architecture. And I'll get into a little bit in a minute on what they were actually looking at. And they were able to look at the boundaries that changed between the different types of cellular architecture and come up with these areas. And you can sort of see, for example, that they came up with these sort of strips and regions within the cortex. And they paired this with stimulation studies. So they were working in collaboration with a neurosurgeon who was stimulating different parts of the brain. And what they came up with were these different labels associated with what was elicited when part of the cortex was stimulated. And again, they came up with strips and regions in the brain that might relate in some way to the structure. And this was some of the first hard evidence that there was a link between structure and function. This is a slightly more recent work from Rizzolati's lab showing a similar thing. So here I've just outlined in pink, using my amazing PowerPointing skills, one region of motor cortex. There's several other sub-territories surrounded here. That's delineated by cytoarchitecture, the cellular architecture. And what you might be able to see is these little open and filled symbols here represent what was elicited by stimulating in the monkey cortex. And what you can see is there actually are some really sharp boundaries here. So here's one in particular where the black is all in one territory and the white unfilled is all in the other territory. So there's some really striking overlap between these things. So what do I mean by cytoarchitecture? Well, I don't necessarily mean that there's one type of cell in one brain region and another type of cell in another brain region, but more that the distribution of different types of cells can differ from one region to the next. And I'm mostly going to talk about cortex today, but of course cytoarchitecture could refer to anywhere. So cortex tends to have how many layers? 6, yes. 6 layers. So generally it has 6 layers. I've outlined them here. And what you can probably see from this diagram is that some of them relate very strongly to inputs, and some of them relate very strongly to outputs, and some of them relate to both. And some of these layers can be incredibly complex depending on what we're looking at. So I'll just show you a couple of examples. I'll show you how it is that we can use cytoarchitecture to do what Brodmann, for example, did here, which is divide up the brain into multiple parcellations, multiple subregions. So, for example, here's an area of the brain that doesn't have a layer 4 at all. So area 4 receives a great deal of inputs from both the cortex and the thalamus. So it's a very important area for inputs. From those other areas of cortex, and agranular cortex doesn't possess a layer 4. It's not discriminable at all. But what it does have, and it does have these other layers, by the way, but they're really poorly discriminated. If I asked you to draw lines where those other areas of cortex were, you probably wouldn't be able to do it very easily. So this kind of cortex really doesn't have great laminar structure. Except for one layer here, which you could probably pick out just from wherever you're sitting there. This is layer 5, and it contains these giant neurons, otherwise known as BET cells, which will have these huge axons. And this is one idea of how structure might relate to function. So these huge axons in these neurons have the ability to transmit information really quickly. And so motor cortex really needs this kind of property in order to be able to trigger movements. It needs giant axons. So here what you're looking at is primary motor cortex with these giant cells, and you're comparing it to premotor cortex, which is directly adjacent. You don't see quite such huge cells in premotor cortex. Here's another example of a different cytoarchitectonic territory, and this is primary visual cortex. And primary visual cortex not only has a layer 4, it has 3 of it. So let's see, I can't quite line up my mouse. Yeah, so 4A, 4B, and 4C here. And you probably could just draw a straight line delineating layer 4 here. It's really, really well delineated. It's collecting a lot of different inputs from a lot of different types of visual subregions of the thalamus. And in addition, with a different stain, so this is the Nissl stain on the left, which is the most commonly used cell body stain. And on the right here, cytochrome oxidase, which reveals that there is even microstructure within the cortical layers. So there's a lot going on in primary visual cortex. And here's another one. This is the last example I'm going to show, which is prefrontal cortex. And prefrontal cortex is known as granular cortex because it has this layer 4, which is populated with many of these small granule-type cells. And again, you can see them here. So area 9 has more of a premotor profile. And areas 46 and 946, you can see this stripe of layer 4 across here. And it's thought that what's happening here with this very, very prominent layer 4 with these large neurons is that it's receiving multiple inputs from other areas of association cortex, which might make it higher level integrative cortex. So we really do care about these things. And I have a whole other talk that I will not give now about why it is that prefrontal cortex is prefrontal and how we can tell and whether this granularity is a key structure of it. But instead, I'm not going to do that. You can ask me about it later if you want. So a lot of that stuff that I showed you was done in single or maybe 2 cases of monkey and human brains. But modern day cytoarchitectonics have allowed us to really get a feel for how variable this is or how reproducible from subject to subject by coming up with these very detailed profiles. So in this study, for example, they first of all separated out cortex from white matter, and then they made it into tons and tons of tiny territories by having strip after strip after strip. And then they mapped the density of neurons in the volume of each of these strips, and they mapped it out, and then they looked for boundaries here using this algorithm that looks for the maximal change between one strip and the next strip. So what they're looking for is distinction between the areas. And that way they can come up with areas, and they can do this across multiple brains very rapidly. And by very rapidly, I mean it takes years and years. And so, for example, here they've parcellated in one human brain, and the panels on the bottom and on the right. And here, this is an average probability map shown across 10 human subjects. And this is actually for quite a variable area. So this is area 10, which is the frontal polar cortex. It's one of the more variable regions. So for each area, we can now not only figure out where it is, but how likely it is to be there in each individual. I think this is really important. No talk on microstructure can go by without also talking about myelo-architecture. So we don't just have cell bodies in our cortex, we also have axons and dendrites, and we have connections. And so if we want to look at our axonal connections, we need to stain for something else. So I already showed you the Nissl stain for cell bodies. You have these little puncta that are appearing. Here we have a stain for myelin, which shows up the myelinated axons very, very darkly. And then this is one of the more famous stains. It's the one that Cajal used when he was first coming up with the theory that there were neurons in the brain that did things at all. And this is the Golgi stain, which stains the entire neuron. A myelo-architecture, we were saying the other day, there are very, very few publications covering it. And those that did are very unhelpful for us because the territories that they came up with when they were looking for boundaries between different distributions of myelo-architecture came up with completely new names that don't refer to the names that people came up with to look at cytoarchitecture. And so it's really hard to pass across them. But one thing that we do know is that these territories do exist, and there are boundaries between them. And I think one open question is how they line up with cytoarchitecture and our other measures of parcellation. But it could be really important. And one of the reasons it could be really important is because it actually seems to line up more accurately with T1-weighted MRI measures than does cytoarchitecture. So T1 MRI is actually not a great measure of cytoarchitecture at all, as far as we know. It seems to reflect myelo-architecture much better. So as we go through cortical depth here in this graph, I'm showing you myelin is in green and the T1 MRI signal is in yellow. And what you can see is that the pink cell body stain really doesn't line up nearly as well as the myelin stain does. So there could be something to this. Which brings us nicely to connectivity. So we already talked about cell types and how they might relate to function, but the other thing that might relate strongly to function is connectivity. And I'm mostly going to talk about anatomical methods that don't use MRI to set the stage for some of the next speakers. Some of the earliest forms of connectivity studies use blunt dissection, and this is really macrostructural, although rather beautiful. So you can see these fantastic fiber bundles which were just produced at the macrostructural level. But if we want to get down to the microstructural level and we want to see connections, not just the big long-range bundle type connections, but also the small local connections and the small long-range connections, then we need to turn to an invasive technique. So I'm talking about track tracing here. And I think that track tracing is still incredibly important. It's still being done. It's invasive, so it can only be done in animals. It involves the injection of a dye, either a radioactive or a fluorescent dye or something else that you can visualize into a brain region, and then waiting for the cell's own active transport mechanisms, the actin and dynein, microstructure, the microtubule structure of the cells, to actually kick in and transport that in one direction or another. So there are anterograde traces, which can be carried from one end to the axons. There are retrograde traces, which can be carried back toward the soma from the cell terminals. And there are traces that go in both directions, so you could get one or the other kind of trace. And these require you to wait a little bit and then actually extract the brain post-mortem and look histologically, slice by slice. So here's an example from a fluorescent one from Suzanne Haber's lab, showing that you can look, for example, at multiple branches of the same tract. This has been reported in ways similar to this, so talking about the territory in which it was injected, and then where it ends up. And if we're lucky, we also get where it went on its way to where it ended up, which is much more rare in track tracing studies. And all of this information, it might be really useful because it might tell us something else about function. So this is a paper from really a long time ago now by Dick Passingham, which was one of the first papers to shape the way that we thought about connectivity from a parcellation point of view. And what he stated was that the connections of an area could form a kind of fingerprint. That is, not one area connects to another one and no other area does, but that the particular pattern of connections might form a functionally relevant module. So here's an example from a couple of areas. So here's area 14 and area 9, which are in quite different parts of the frontal lobe. And what you can see is that they're connections with other Brodmann areas. So these are Brodmann area labels here. There are also other areas of the frontal lobe. He cared a lot about the frontal lobe. And what you can see is that this one has a pattern of connections mainly to these regions, and this one has a pattern of connections that's much more homogeneous. And what he did is he divided up these types of connections along a couple of major axes. And he was actually able to show that there were different territories within here that mapped well onto other territories that have been defined with cytoarchitecture. So I'm showing you here lateral regions, orbital and medial regions, more dorsal regions, more dorsomedial regions, for example, all sort of mapped away from each other. Now, I've described these in terms of where they are on the cortical surface, but these regions also have really different cytoarchitecture. So there seem to be multiple ways this can line up. And so if you got a lot of connectivity fingerprints, then it might be possible to find out something else about the function of those regions and how they differ from each other. And this is a relatively extreme example of this, looking at the difference between the orbital and medial frontal lobe connections, showing that they really have a very, even just by eye, you could tell me that these are two separate networks. So I think this is kind of a really important aspect. And as I said, if you try and do this in humans, well, you have to wait for someone to donate their brain, and then you can put a little bit of fluorescent dye in, and you can wait for it to travel, and it will travel just a few millimeters, and you'll only get local connections. So my case for that is to set the stage for needing something in the human brain, such as diffusion imaging, which is going to reveal whether this is the same in humans as it is in monkeys. Okay, but I really want to talk about the relationship between these things and how we can assess the relationship between them. So I've talked about these multiple ways that we can use to parcellate the brain. Is it possible that we could use these ways to parcellate the brain in a way that is meaningful no matter which measure you use? And I think that is possible. There's some beautiful work from Helen Barbas's group showing that cytoarchitecture predicts connectivity really strongly. So what she did was she divided up the brain into these different types of tissue, going from agranular to dysgranular to these two types of eulaminate cortex. So the darker colors, as you can see, have way less differentiation, they have way fewer connections, and the lighter areas here have much more of that laminar, granular type structure that I was talking about before. So she divided up the brain into these areas, and then she made predictions about which areas would project to which, based on the fact that, I showed you those 6 cortical layers at the beginning, and some of them have more inputs and some of them have more outputs. So based on what we would predict about those different cell types, how would we predict these project? And what she came up with was the idea that those darker areas are more likely to project to the lighter, more differentiated areas. And when you actually look at those connections using track tracing, that's exactly what you find. So the prediction holds up. So cytoarchitecture predicts connectivity. This has also been shown more recently. This is some really beautiful work. Martin van den Heuvel is the first author on this paper. So this is looking at cytoarchitectonic areas. It's looking at areas delineated by neuron size. And it's looking at macroscale connectivity as measured by diffusion imaging. And what they found was that the neuron size in layer 3, which is very, very strongly correlated with macroscale connectivity. So it seems to be a very, very good predictor of what these long-range connections are going to be. And if I went back to that slide, which I should have put back in again, that is what layer 3 is doing. So layer 3 has the long-range connections emanating from it. So this seems to be very strong evidence that there's a link between them. I also wanted to go really, really micro for a minute and talk about dendritic architecture. So dendritic spine density is a really important measure of microstructure that has received a lot of attention recently. So we've talked a lot about axons and dendrites and cell body types, but in addition to this, there seems to be a very, very flexible, almost fast-moving component to cortical and subcortical circuitry, which is what the dendrites are doing. And the dendrites, so there's a number of measures that we can come up with here. So here's a Golgi-stained neuron. I showed you that stain before. And here's a schematic of it showing that it has dendritic lengths. So some of these are longer than others. There are dendritic branches. So there are branches coming off of here. And there are dendritic spines. And there are different types of dendritic spines. There are thin ones, and there are stubby ones, and there are ones called mushroom-shaped spines. And these appear to be correlated with synaptic density. So it seems as if when you're forming dendritic spines, which you seem to be doing on a dynamic basis, you might be forming new, more permanent connections as you move from these skinny type spines to these fatter type spines. And what happens is if we map out the spine density, for example, in layer 3 of different cortical regions, this also, so if we do this, and we also take the macrostructural white matter diffusion imaging connectivity and functional connectivity, and we correlate it with things like spine density, the length of the dendritic tree, what else do we have up here? I can't read this, the segment length, and so on, then we get this very, very strong correlation between measures of connectivity on the long range and this very, very small scale microstructural connectivity to do with the very, very local connections. Now, what this means we're not quite sure of, but it does give us maybe a powerful tool for understanding what's happening in the dynamic brain from time point to time point. And before I go into that, I just want to add that although the Golgi stain is amazing, actually it doesn't capture the whole dendritic tree. And another plug for animal studies here is that the only real way to see the full complexity of the dendritic tree and to see spines on those very far dendrites is to fill cells post-mortem with lucifer yellow dye. You can actually do this in the post-mortem human brain as well. It takes a long time to do this, and then you can image all the way to the ends of these neurons, and you can image spine density much more accurately. And this is the one bit of work from my lab that I'm going to show today. And this has been shown in rodents, for example, to correlate with differences between groups in grey matter volume. So what they did here was they exposed animals to an auditory fear conditioning paradigm and compared them with a control group. And they parcellated out using anatomical features, gross anatomical features being very, very reliable from rodent to rodent. This is the rat brain. No, I'm lying. This is a mouse brain. I'm sorry. So they're very reliable. They parcellated out these regions manually. They looked for differences between the groups in grey matter volume. They saw larger grey matter volume in areas related to fear conditioning. And then they imaged the dendritic spines. Actually, you don't have to fill the neurons if you have a mouse that has fluorescently labeled neurons. And you can already see by eye that the controls on the left compared with the fear-conditioned animals on the right, the fear-conditioned animals had more spines in those areas. And they showed a strong correlation between dendritic spine density in these layer 3 neurons and the grey matter volume across the groups. And that didn't correlate with things like neuronal density. So it explains what we're measuring maybe when we're measuring changes in grey matter volume from animal to animal. And so it turns out that if you create a model, such as this one, looking at which aspects of macrostructure and microstructure, so some of the examples that I've shown already, predict connectivity, it seems to be a combination of things. And so what they came up with was when they took into account both macrostructure and microstructure here, they came up with the possibility that both neuronal density and the Euclidean distance between areas might be a predictor of how well they connect and how well they communicate with each other. So this story is still very much evolving, and I hope one thing that I might have convinced you of is that cytoarchitecture and histology and microstructural measures are not old technology, and they're not out of date. They're directly relevant to all of the questions that we're asking today, and it'd be really great if you guys can bear that in mind as you hear the other talks. Having said that, there are a few limitations of microstructure. It can be really time-consuming to do. Requires a lot of dedication. It takes a good couple of years to do a study on a single brain sometimes. Usually it can only be performed post-mortem, which means that we have fewer samples or we're limited to animals. And we're still figuring out the relationship, which could be very complex, between structure and function, and between the MRI measures that we're going to hear about today. So that's it from me. Thanks very much.
Thank you very much for this wonderful talk. We have time for a few questions. So if you want to ask questions, you get closer to the microphone. Yes.
Morning.
It was very nice talk. I have a question about the cytoarchitecture. So when I always wonder, because some of the regions like V1 are very, very clear and laminar, and some of the regions, gray level index really looks very continuous across layers. And I'm curious how people decide where to put the labels for the layers.
Right. Yeah, and I had a bit of a mental breakdown at some point when I was doing this, when I was trying to look at some of the papers and I couldn't tell by eye the difference between one region and the next. So I think that in the past it just relied on someone having a great deal of expertise and being able to see that. But what's kind of nice now is that some of the measures that I showed where they had stripped the cortex down and they had come up with this this gray level index that you had talked about, which is kind of a measure of neuronal density in some ways. What they're looking for is not things within the area that make it similar to itself, but where the boundaries lie, where it becomes sharply different from another area. And they've kind of come up with a way of doing that is more independent of a sort of observer expertise in some ways. But I think the short answer is that it's not perfect. And there are a lot of claims that it's very, very independent and objective, and I don't really know if that's the case for some of the past studies. So I think it's important to apply those new methods going forward. And a couple of groups, like Carl Zillis' group and Katrina Munt's group, have been really applying this to multiple brains. And one of the questions is, if you took my brain and your brain, can we even find the same areas across them? And is there a way we can do that reproducibly? So yeah, I think that's a great question.
Thank you.
Wonderful, very, very inspiring talk.
Thank you.
But this is a question for the whole panel, really. Will this seminar cover other areas of the brain besides the cortex? Anything deeper in the brainstem or in the basal ganglia?
I know that was a bit rude of me to only talk about cortex, wasn't it?
You do.
It's not in my talk, but if you look up a paper by Jean in 2008 in cerebral cortex, you'll see a comparison of functional connectivity with track tracing by diffusion things. I can, in other words, I should have put it in my talk. There's track tracing versus functional connectivity, corticothalamic. So there is such a thing.
Thank you.
I have one slide where I'm showing just one slide. Okay, I have a question. So when we look at cytoarchitecture, the motivation has been to divide the brain in different areas, but I was wondering whether you can have a repetition of the same cytoarchitecture in distant area in the brain, you know, not joint areas, like one in the frontal and one in the parietal, for example, can have maybe the same cytoarchitecture, and if it has been reported in the literature. Do you have any idea?
I didn't come across anything in the literature that reported that, and I actually wouldn't be surprised if that was the case. I think that it's likely. Because what we were just talking about with the first question there was what are we basing it on? And we're really basing it on boundaries, right? So I think it is possible. That would actually be a great study, I think, to compare one region that's distant to another one. But yeah, if you think about the relationship between structure and function, any higher order association cortex, for example, might have that kind of structure with a complex granular layer 4, and look quite similar when we put them together. So yeah, I don't know if anyone of the other speakers knows of this study.
Well, it makes me think of sort of how, lightly myelinated anterior cingulate cortex, it's also agranular, maybe similar, it's also very thick, just at very coarse properties we can measure with MRI, to anterior insular cortex. They're on opposite sides of the brain, but they've sort of been described as being similar, at least in the gross measures of architecture. So that would be an example.
Yeah, that's actually a great example. And they both have von Economo neurons, of course, as well.
Right.
Yeah.
I have a question. For the Barres and Helga's work, they use spine density as to represent this cytoarchitecture. But for other work, they also use segment length or other measures. Maybe because they use a different measure because the limitation of the work at that time. But also there's another possibility that these different measures, they have certain relationship among each other. I wonder, would you like to give us some hint about these parts, like make some example about you think the possible relationship between these different measures of cytoarchitecture, maybe?
Yeah. So I think that's still being looked into, and I'm not yet an expert on those different measures of dendritic complexity. But it is true that if you look, if you're comparing groups, so for example, some of John Morrison's work looking at the aging brain and looking at what he calls synaptic health, he's looked at the different measures like different types of dendritic spines, so he's subcategorized them, and then dendritic branching and the segment length. And what they found in the aging brain, in prefrontal cortex, just to talk about another cortical area, is that it's the thin spines that seems to be most correlated with performance on things like spatial working memory in aged monkeys. And so I do think that that's one example where we shouldn't lump together all of those measures. And I know I showed a couple of studies that do kind of categorize them all as one thing. I think they are very different things, and they take different amounts of time to develop. So they probably have impacts on plasticity and degeneration that are very different from each other. And I'm really looking forward to work, and we're starting to do some of that work in our lab as well, showing us more about that.
Thank you.
Thanks.
Say
We
a
have-
little bit more about the work that you're doing now on filling the cells to show the full dendritic tree.
Yeah, absolutely. So one of the things that we've become interested in our lab is what we're measuring when we're measuring these changes in things like grey matter volume and also changes, I didn't talk about this very much for time constraints, but we're also interested in what some of these changes like fractional anisotropy changes in the white matter relate to. So what we've been doing is taking monkeys that were trained on tasks that were dependent on different cortical subregions. So a task that was very prefrontal cortex dependent, and a task that's very dependent on the perirhinal cortex, an object recognition memory task, and a task that causes the monkeys to form a learning set, which are temporal lobe and frontal lobe dependent, respectively. Measuring changes in cortical thickness and FA, and then filling the cells from, in this case, the ventrolateral prefrontal cortex. So it's basically a sort of slow and steady technique where you have to use a DAPI stain to find the cell body of the cell. You have to stick an electrode into it and then very, very slowly infuse the lucifer yellow dye and just wait until it's gone to the very extremities of the neuron. And this is actually really hard to do in the monkey because monkey neurons are big and they pass through multiple layers of cortex. And so if you take a brain slice, it's likely that that neuron will leave the brain slice at some point and its trajectory elsewhere. It's easier to do in the rodent brain, and there's more literature on it. But I think it is quite important because the MRI scanning that we're doing in monkeys is more similar to humans. We're trying to understand what the relationship between those things is. And again, for us, it looks like the spine density measures seem to be very well correlated with cortical grey matter differences between groups, maybe more so than the dendritic branching and complexity. And that's probably because we're using a cognitive task training paradigm, which doesn't result in such dramatic changes.
The dye has to be injected when the monkey's alive.
No, post-mortem. Yeah, so you could do this in the human brain, and actually it has been done in the human brain. So there are a couple of ongoing studies that I know of, taking, for example, tissue from patients with epilepsy that's removed during surgery, and looking at the properties of neurons in the healthy tissue and in that surrounds the area with the damage, and then in the focus area. So this is actually one example of a technique that could really span species and probably should be used across species. I think maybe
Yeah, I think
should
we should
move on to
probably
the
move
next
to the
talk.
next one. Thank you.